Hi everyone, let's continue our discussion from the last time where we were talking about sequence spaces and fitness landscapes. We will continue from where we had exactly left off last time. We are considering a sequence space associated with DNA of length L, and we had calculated that there will be 4 to the power L nodes, each representing a particular sequence of length L. And we were interested in calculating the number of neighbors that each of these 4 to the power L nodes has, and a neighbor being defined as that sequence which varies from one particular sequence at one nucleotide only, and it's identical at the other L-1 positions. So just to recap, we are going to represent our sequences as nodes. This is node number 1, 2, node number I, and eventually going all the way up to node number 4 to the power L. And we are connecting node 1 and node I if sequence associated with node 1 varies with sequence associated with node I at exactly one nucleotide only. If that's the case, that's when we are connecting node 1 and node I on our graph, and this gives me a connection between various nodes associated with this DNA sequence of length L. And then we had moved on to talk about how many nodes or how many neighbors does each node on this graph have. And the answer to that was 3 to the power L, because for every position in this DNA sequence of length L, the variation between a node and its neighbors could be at any one of those L positions. If the variation between the neighbor and the node itself is occurring at position number 1 of the sequence, then it could have 3 different nodes. Suppose the node that I am talking about has nucleotide A on the first position. The node that varies on the first position only could have a T, could have a G, or could have a C on position number 1s. Hence, sequences that vary only at position number 1 are 3 in number. Similarly, if I am talking about sequences that vary only at the ith position in this DNA sequence of length L, I am going to have 3 neighbors associated with that. And when I sum all these neighbors up from nucleotide position number 1 to position number L, I get 3 times L, and that gives me the relationship that every node on this graph has 3 L neighbors, right. So what that tells me is that the total number of nodes that corresponds to the total number of sequences which are possible of length L is 4 to the power L, and each node has number of neighbors equal to 3L. So now what I want you to think about is that how densely is this graph connected with each other. Given a number of nodes and edges, how many nodes of the total number of nodes that are available in this graph, what fraction of nodes is every single node connected to? So the total number of nodes is 4 to the power L, but every node is connected to 3L other nodes. That means the fraction of nodes as neighbors so of all the nodes that exist in this graph, what fraction of nodes are a neighbor of any particular node I, and that will be 3L, which is the actual number of neighbors divided by total number of nodes 4 to the power L, and we can subtract 1 because 1 is that node itself. So, if I am what we are doing here is that if I am talking about node I, there are lots of other nodes in this network, and the total number of nodes, if I leave aside the ith node, the total number of nodes is 4 to the power L minus 1, because I am not counting that node itself, and out of these 4 to the power L minus 1, this node is only connected to 3L number of nodes. So, the fraction of the nodes that every single node is connected to is given by 3L divided by 4 to the power L minus 1. And now, what I would like you to do is perhaps pause the video for maybe 30 seconds or so, and then try and plot this graph and see what you expect this behavior to be. As the sequence of the DNA that we are talking about increases, so x axis represents increasing length of the DNA sequence that we are talking about. The y axis is what is the fraction of nodes as neighbors that every single node in this graph has. So, on y axis I want you to plot 3L divided by 4 to the power L minus 1. So, perhaps you can just pause the video for 30 seconds or so and think about this problem for 30 seconds or so, and come up with a very qualitative estimate of what you would expect intuitively this relationship should look like. All right. So, what we can see here is that when L is equal to 1, that means we are only talking of DNA sequence of length 1. This is 3 divided by 3. So, this is 1. And very rapidly what you should expect, and you can work out the numbers for L equal to 2, L equal to 3, some of the smaller DNA sequences can be worked out by hand. What you would note is that it decreases very rapidly and approaches 0. So, what this tells us that our graph is very sparsely connected. For any reasonable length L, that means the total number of nodes which are present in the sequence, every single node is connected to only a very small fraction of the nodes other than itself in the total number of graphs. So, this graph, the sequence space associated with this DNA is a very sparsely connected graph. All right. So, now what we are going to do is imagine that this DNA sequence that we are talking about, which is a sequence of length L, corresponds to the DNA sequence of a organism, and for the sake of simplicity, let us say bacteria. So, this L corresponds to DNA sequence of a bacterium. Obviously, if length of the DNA polymer that we are talking about is L, that means we are talking about a graph which is 4 to the power L nodes. Maybe let us just draw a small section of this graph, and let us say these are 6 nodes on this graph. Let us number them, call them A, B, C, D, E, F. And again, consistent with what we have been defining so far, each one of these nodes represents a particular sequence. Now, not all are going to be connected with all graphs. So, we are going to establish these connections, and a connection would mean that you can go from one node to another by acquisition of a single point mutation. And if that is the case, let us define these relationships to be of this sort. Let this be the graph that we are talking about. So, you can see connections between some of the nodes, but not connection between all, but all possible connections do not exist on this graph. Because L is a DNA sequence of a bacteria, every sequence is going to have a corresponding growth rate associated with it. If I had a bacterium whose DNA sequence corresponded to that of the sequence which is at node 1, that bacterium would have a growth rate associated with it. Should there be a point mutation and the sequence from A goes to sequence at B, this bacterium which is carrying sequence B would have a growth rate associated with the sequence corresponding to that of node B. So, let us write down the growth rates associated with each of these sequences, and I am just going to make up numbers so as to illustrate the characteristics associated with these fitness graphs. These are not representative examples, but I am just making up numbers of growth rates which could perhaps exist in a graph such as this. So, let us just write down the growth rates associated with each particular sequence, that is each particular node that we have in our graph. Alright. So, let us say sequence A grows at 1.5 and the units for the growth rate will be time inverse, we are not specifying units. B could be maybe 0.8, D is 3, F is 2, C is 1 and E is 0. So, these numbers represent the growth rates associated with the sequence that each node is representing. Now, let us imagine that we have a sequence, we start off our experiment with a genetically identical bacteria and all bacteria have sequence which corresponds to node C. Okay. So, what that means is I start off my experiment in a test tube and I add a few bacteria to this tube and all of them have the exact same sequence as node C. So, genetically all individuals are identical, which means this is where I am starting my experiment from. Now, what is going to happen as this experiment plays out in future is that these bacteria will keep on dividing and eventually one of them will acquire a point mutation. For the sake of simplicity, for the time being we are only talking about point mutations, we are not talking of deletions, duplications, etc. or acquisition of DNA horizontally, we are only talking of single point mutations. One of these bacteria upon division is going to acquire a point mutation. So, let us see what happens, what is going to happen here when that happens. Because mutations can happen randomly and in this graph the 3 point mutations which are possible are C going to B, C going to F and C going to E. What happens if one of the bacterium in this culture acquires a mutation and goes to sequence E, for instance? Now, the growth rate associated with sequence E is 0, which means this mutation that has occurred here, the resultant bacteria does not grow at all, it never divides, which means that the mutation that has happened is what is called a lethal mutation. That means the bacteria which is carrying this particular genotype would not be able to survive, hence its growth rate is equal to 0. So, one of this mutant is not going to survive and it is going to get eliminated by selection and the population still remains at node C. Then you may have another mutation occurring and one of the individuals in the population could go to sequence B. What has happened here is that while the rest of the population is growing at growth rate equal to 1, this one mutant which has sequence corresponding to that of node B is growing at a growth rate 0.8, which is less than 1. So, the growth rate or fitness of this bacterium is less than the parent genotype that it came from, and when there has been a decrease in fitness or growth rate, this mutation will said to be a deleterious mutation. So, you could have lethal mutation, you could have deleterious mutation. What could also happen, the third possibility as shown here is that mutation from C to F might happen and this results in increase of fitness from 1 to 2. And when this is said to happen, this type of a mutation is referred to as a beneficial mutation. And now when there is a mutant which belongs to genotype F, that mutant grows faster as compared to the original genotype that the rest of the population is located at, and hence because of selection that mutant starts eliminating the individuals of the parent genotype, and the population eventually moves from point C to point F. The relative frequency of individuals which belong to genotype C decreases, and the relative frequency of the individuals which belong to genotype F increases, and eventually all L and N individuals come to sequence F. And when we are here, the whole story repeats itself. From F you could go back to C in the graph that we have been given. From node F you could have a mutation and go back to C, sequence C, but that entails a decrease in fitness. Hence this is a deleterious mutation and will be eliminated by selection. So this does not work, but you could have a mutation going from F to going to node D, which entails an increase in fitness from value of 2 to value of 3, and then you will have movement of population approach D. So when we are talking of mutations, you could have these 3 type of mutations. There is another type of mutation that you could have happen in the process, which is, we draw another node here, which is, let us say, C going to node G, and the fitness corresponding to node G is just 1. So should this mutation happen, you have an individual in the population which is also growing at the same rate as the parent's genotype, but that genotypically this mutant is distinct from the parent genotype that it came from. So these are genotypically non-identical but phenotypically identical, and when you have a mutation that has happened which does not lead to a change in the fitness associated with the growth of the organism, this type of a mutation is said to be a neutral mutation. Okay. So again you have this graph, and you are going to have movement of populations on this graph depending upon the fitness values associated with particular node. When you have movement from a node to another node which leads to an increase in fitness, that means selection is going to act and is going to eliminate the individuals which belong to the parent genotype, and you are going to have population, more number of individuals present in the new genotype which is growing at a rate higher than the original genotype. On the other hand, if the mutation that has occurred is deleterious in nature or lethal in nature, then again selection is going to act and eliminate those mutations. Neutral mutations, which are mutations which lead to individuals which are genotypically distinct but phenotypically identical in the sense that they are growing at the same rate, have a very different implication on evolutionary dynamics. That is something that we will come back to later in the course. For the time being, I want you to remember, I want you to sort of realize the significance of these 4 type of mutations that can happen, and how in this sequence space and the graph like structure that we have defined here, populations are going to move along channels where fitness is increasing. All those paths where fitness is decreasing are not going to be allowed because of selection. Right. Okay. I want to emphasize a couple of other points related to this graph. The slide is quite messy, so let me just redraw quickly this graph again. So we have node A, B, C, and the corresponding fitness numbers are 0.8, 3, 2, 0, 1, 1 and 1.5. So what we realized is that if our starting population is node C, then what is going to happen is that population is going to move towards node F first, and from F, once all individuals Selection will dictate that individuals belonging to genotype C get eliminated and the mutant which is at sequence F grows faster than those at sequence C, and hence population will move towards F. Once you have large number of individuals here, you wait for a mutational event so that one individual with genotype corresponding to D arises, and hence you have population moving to genotype T, again via action of selection taking place here. So, from C, this is the direction that the population will move towards and eventually end up at node D starting from C. We can see that this ensures that the population ends up at a node which has the highest fitness among all nodes. Okay. And which makes sense because natural selection would mean that you are moving towards sequences which grow faster and survive better than any other sequences that might be competing with them in the environment. But what happens, let us say if the fitness of node A was not 1.5, but the fitness of node A was let us say 5. Now, in this graph that we are talking about, what is going to happen is that there are a few things that play out. One thing that we can clearly note is that D is no longer the highest fitness among all nodes. If this changes, this is no longer true. And now what we have, that node A is the highest fitness, and for that reason this is called the global optimal in terms of growth rate associated with bacteria. So, this is the global optimal value of growth that individuals which are carrying one of the sequences defined by these nodes can hope to reach. But we realize that if we are starting from genotype C, to get to A, we have to take a step down in fitness and then take another step up in fitness, going up to value of 5. But this step down in fitness is not going to be facilitated via natural selection. So, this in a strict Darwinian sense is not allowed, and hence the population does not quite reach A, but will take these two steps up and end up at node D. What is the characteristic of node D is that the fitness associated with node D is greater than fitness of any of its neighbors. So, D has two neighbors here, B and F, and the fitness corresponding to node D is more than that of fitness corresponding to node B or node F. Hence, this result holds for node D, and whenever that holds, we said that that particular node is a local optima. And local optima will have consequences in terms of how populations move on fitness landscapes, something that we will come to a little bit later on. Right. So, we have global optima and we have local optima on this graph. All right. Another curious perspective that is associated with this graph is what happens if the starting genotype of our population was not node C in this graph, but was actually node B. What happens if it was node B that we were starting with? Now, node B, remember, mutations are random events. Mutations take place randomly. There is no directionality associated with where and how mutations are going to take place. So, starting from node B, we do not know a priori if the first mutation that is going to happen in this test tube where I have genetically identical cells of genotype B growing, I don't know a priori if the first mutation that's going to arise in this tube is going to be a mutation towards sequence A or mutation towards sequence C or mutation towards sequence D. These are all presumably equally likely, but this has important implication in terms of where the population ends up. If the first mutant that arises here corresponds to sequence A, that means the fitness of that mutant has gone up to 5 as compared to fitness of 0.8, which was that of the original genotype, which is a big increase in fitness, and hence selection will act and the population moves towards 5, and the population has reached the global optima. It cannot do better than growth rate of 5 at any of the nodes, hence the population stays there. However, if the first mutant that arises was this mutant C, then the population fitness has increased from 0.8 to 1, selection acts and the entire population moves towards the sequence corresponding to node C, and from there on the story repeats itself. From C it moves to D, from F, and from F it moves to eventually D and stays there because it is stuck in a local optima. Or it could happen that the first mutation is from node B to node D, and in that case it's immediately reached this local optima and the population stays there. So, depending on which was the first mutation that happened in the environment, the dynamics of evolution of these populations are going to vary, and in that sense this evolutionary dynamics of population growing in this test tube is being played out by chance, which decides which is the first mutation that happens in a test tube, and that eventually decides the movement of the population on the fitness landscape structure that we are talking about. So, we'll stop this lecture here and we'll continue our discussion on fitness landscapes in the next class. Thank you.